RNA extraction. RNA extraction overview. What is RNA? Ribonucleic acid, a nucleic acid present in all living cells. Its principal role is to act as a messenger, carrying instruction from DNA for controlling the synthesis of proteins. Although in some viruses, RNA rather than DNA carries the genetic information. How many methods of RNA extraction? There are three major techniques extensively used for RNA extraction. Organic extraction, such as phenol guanidine isothiocyanate based solutions, silica membrane based spin column technology, and paramagnetic particle technology. One of the most commonly used methods is the phenol guanidinium isothiocyanate based organic extraction. RNA extraction method. Guanidium thiocyanate phenol. Step 1. Sample such as cultured cell tissues etc. Sample is first homogenized to release RNA out of the cells. Step 2. Homogenize in denaturing solution. Guanidine thiocyanate. Guanidium thiocyanate denatures and precipitates protein, includes RNAs, which degrades RNA. Step 3. Add sodium acetate pH 4.0. Acidic pH selectively denatures DNA but not RNA. If pH remains 7, both RNA as well as DNA got denatured. Step 4. Add water saturated phenol solution. Water saturated phenol forms two layers upon mixing, above aqueous layer and lower organic nonpolar layer. Because protein and DNA are already precipitated, they partition into layer nonpolar phenol layer, and along with lipids only RNA remains in upper aqueous water layer. Step 5. Add chloroform and isomile as 24:1. Chloroform clears the fuzziness between aqueous and organic layers. Isoamyl alcohol prevents forming of phenol due to vigorous shaking. Step 6. Soap add isopropanol. RNA present in upper aqueous layer is collected after centrifugation and precipitated by isopropanol. Mix, incubate for 30 minutes at -20 degrees centigrade and spin. Step 7. Pellet, add 70% ethanol. Precipitated RNA is washed 3 times with 70% ethanol to remove any phenol or salt that may be present. Again, mix, spin and repeat this process 3 times. Step 8. Drying the pellet. Precipitated RNA is dried at room temperature for 3 to 5 minutes. Step 9. Dissolve in RNase free water and store. RNA is dissolved in RNase free water and used for further experiments are stored at -20 degrees centigrade. Functions of different reagents used in guanidium thiocyanate phenol method. Guanidium thiocyanate. Cheotropic agent denatures protein including RNase and nucleic acid binding proteins. Acidic pH. Sodium acetate denatures DNA but not RNA and partition it into organic nonpolar phase. Saturated phenol. Nonpolar solvent collect lipids, precipitated protein and DNA. Chloroform clears demarcation between organic and aqueous layer. Isoamyl alcohol prevents forming of phenol due to mixing. Isopropanol precipitate RNA by lowering dielectric constant of water. 70% alcohol removes any salt or organic solvent that exists with RNA. Thank you very much.